Perfect. So my name is Agnieszka.
Yes.
I'm an assistant professor in the Department of Pediatrics in our Division of Hematology, Oncology, Stem Cell Transplantation, and Regenerative Medicine here at Stanford University School of Medicine. And today I'm going to be speaking about Fanconi anemia and gene therapy, as well as other new treatments that we're developing here at Stanford. So Fanconi anemia is a complex inherited multisystemic disease caused by defects in one of 23 different genes involved in the Fanconi complex, which leads to hypersensitivity to DNA damage, causing interstrand crosslinks. This can be associated with many different issues, including congenital abnormalities, bone marrow failure, and cancer predisposition. And as you see here on this slide, the Fanconi complex is quite complex with many different genes that are involved, although a majority of patients have issues in the FANC-A gene, with 60 to 70% of patients having issues in that, 10% having issues in the FANC-C, and 10% in the FANC-G, and the rest being very diverse mutations in diverse genes. And on the right side of the slide is a schematic just displaying the many different issues that Fanconi anemia patients can have, although again, the most common is bone marrow failure and a predisposition to cancer, with an over 1,000 increased risk of malignancy compared to other patients. As I mentioned, progressive bone marrow failure is quite common in these patients, with 80% of them developing bone marrow failure by the time they're 12. And unfortunately, they also have a high predisposition to hematologic malignancies and solid tumors, which is displayed on this chart as well. And while this is really a systemic disease that affects all cells and all tissues, it's these hematologic issues that tend to cause the most problems, especially early on for patients. Because of this, many patients progress to needing hematopoietic cell transplants, which can be curative for the hematopoietic issues associated with the disease, specifically the bone marrow failure. But they are currently associated with pretty significant short-term and long-term toxicities, especially for the about 80% of patients who do not have HLA identical siblings. So while we're very proud that as a field working together, we have found ways to really improve the efficacy of these transplants, as can be seen by the survival curve here on the left, there are still significant issues for these patients. There are still significant 100-day transplant-associated mortalities, especially for the patients who need to use alternative donors, graft versus host disease for many of these patients, infections, and other short-term toxicities. And then additionally, patients unfortunately develop a even higher increased risk of solid organ malignancies post hematopoietic cell transplantations, especially for those who have developed graft versus host disease. And this can be nicely illustrated here on the right from a figure that was taken by a study by Blanche Alter's group at the NIH, showing that all FA patients have a pretty high predisposition to cancer, but especially those who have proceeded with hematopoietic cell transplants, which were necessary for their bone marrow failure. They developed solid tumors much earlier in life. And unfortunately, these solid tumors are incredibly difficult to treat because patients are not cannot easily tolerate our current treatments for cancers due to their DNA repair deficiencies. And so it's these high rates of complications and secondary malignancies and that cause most issues for our patients. And in part, this is due to the chemotherapy and radiation that is used in our transplant protocols today, which is particularly challenging for patients with DNA repair deficiencies. And so it's been our goal and the goal of the field to really try to help these patients in newer ways to eliminate the need for treating them with genotoxic allogeneic transplants by developing alternative approaches. And so here at Stanford, we're working on two specific trials that we think can be very beneficial for patients in different parts of the disease process. For patients that are coming to us pre-development of bone marrow failure, we've been working together with our partner Rocket Pharma and our colleagues in Spain at CIMET and other places now to develop a lentiviral gene therapy for patients with subtype A, with the idea being to identify patients as early as possible in their disease process prior to developing bone marrow failure, take out their hematopoietic stem cells, replace them with gene-modified stem cells by adding in the FANC-A gene using a lentivirus. And then for patients who are already in bone marrow failure or present with more unique subtypes for which gene therapy is not available, instead working to treat them with improved allogeneic transplants using manipulated grafts that have a decreased risk of graft versus host disease, and replacing the total body radiation or genotoxic chemotherapies that we use in these regimens with other therapies. Today, I'll be telling you a bit about both of these trials that we're running here at Stanford that are supported by our Center for Definitive Curative Medicine. We think these are two very innovative trials that we've been working hard to get off the ground and are fortunate to have treated patients that are doing well, and with time hope to expand these therapies to many other treatment sites, including those all throughout the world. There are other treatments that are also being explored for Fanconi anemia, so I just listed them on the slide here, including quercetin, metformin, eltrombopag, and FP045. Each of these therapies is intended to try to stabilize hematopoietic cells prior to a development of bone marrow failure, or could be used post-transplant as well. So the first therapy that I will tell you about today is our gene therapy, again, through a trial that is sponsored by Rocket Pharma. This uses a therapy known as RP-L102, which again is a gene therapy where a functional FANC-A gene is inserted into autologous CD34 positive cells from FA patients. These gene-modified cells can then be transplanted into patients, and this has been shown to be possible without the use of genotoxic conditioning. This initial program was started by the CMET group and was nicely published on in this paper by Paula Ria et al. In Nature Medicine on their early results. And then in collaboration with Rocket Pharma, they have commercialized this manufacturing process to be able to make it more robust and then to be able to administer it to patients throughout the world. This process involves mobilizing our patients with G-CSF and plerixafor, apheresing them, isolating the CD34 positive cells, which again are very rare in these patients due to the development of bone marrow failure. And there's some interesting biology even further showing lower levels of CD34, which makes it even harder to collect these cells. Then these rare sensitive cells need to be transduced with the therapeutic vector, with the lentivirus that again has been optimized to be able to be used on rare and sensitive cells. This manufacturing process has to be done incredibly quickly, and then the cells need to be infused into patients fresh, as there have been challenges that have been noted in the field with cryopreserving Fanconi anemia cells, again due to their sensitivity and being prone to DNA damage and apoptosis. And so these are then infused into our patients without conditioning fresh, and then our patients have been followed here. This is a trial that is now global at multiple sites throughout the world, in particular in Spain, in the UK, and here at Stanford in the United States. Here are the eligibility criteria for our trial. Again, patients have to have Fanconi anemia with complementation group A, as we are giving back the FANC-A gene, and they have to have sufficient CD34 positive cells in their bone marrow, which is determined on a aspirate to be able to have a robust collection to then be able to have the hope of gene correction and stabilization of their disease. As of right now, we are excluding patients who have available and eligible HLA siblings or those who have MDS, leukemia, or other cytogenetic abnormalities or mosaicism, which is thought to likely stabilize their disease. And then we're monitoring these patients in many different ways. Clearly, safety has been important in our phase 1/2 trial, but we're also looking at efficacy by looking at engraftment of gene-corrected cells based on vector copy number, phenotypic correction based on resistance to DNA damage, and then prevention of bone marrow failure and stabilization of blood counts, as well as many other exploratory corollary studies to show stabilization of the marrow post this type of gene therapy. As of the last data cutoff date that was reported of April 4th of 2022, 12 patients had been treated with this RPL-102 gene therapy, again, in our global studies. 9 of these patients have been treated for longer than 12 months. And so this is the data that we're reporting here in the patients that have longer follow-up. All of these patients were fairly young children under the age of 6 in order to identify patients with robust marrow prior to development of bone marrow failure that could be collected, modified, and then returned to them. As you can see here, we've had nice transduction efficiency with the optimized viral vector and transduction system, showing a nice vector copy number in liquid culture and in CFCs with nice transduction efficiency and resulting CFC MMC resistance. And then excitingly, in 6 of the 9 patients that we have 1 year or more of long-term follow-up data, you can see beautiful increases in their vector copy number in both the peripheral blood and in the bone marrow that are continuing to increase with time, thought to be due to the competitive advantage of the gene-corrected cells versus the fragile, uncorrected Fanconi anemia cells. And then not only are we seeing vector copy number and signs of gene correction, but additionally we're showing that functionally these cells are robust and resistant to DNA damage. In 5 of these patients, when we have taken their bone marrow cells post-treatment and exposed them to mitomycin C, which is a DNA damaging agent, we've seen now resistance, which was not seen in these patients prior to treatment. And then excitingly, in the patients with mitomycin C resistance, we have seen stabilization in their blood counts in all 3 lineages. These patients are being closely followed to ensure continued stabilization and that they have no other issues. And we're continuing to follow the other patients that have been treated with shorter follow-up as well. As I mentioned, safety is quite important to assess in this innovative phase 1/2 study. Unfortunately, all patients have been doing quite well. They have been treated without antecedent conditioning, which makes this type of treatment much easier to tolerate. So our patients are only in the hospital for about a week and a half. They don't get the same type of sick as patients undergoing gene therapy for other diseases with genotoxic conditioning or with allogeneic transplant. Our patients have not shown signs of dysplasia, clonal dominance, or oncogenic integration. And we did have 1 patient who unfortunately did not show a robust gene correction early on, who then subsequently developed a viral infection that was unrelated to treatment that tipped her into bone marrow failure. This patient did then proceed to an allogeneic transplant post the gene therapy treatment with no issues, showing that if that does occur, this type of treatment does not increase risks of an allogeneic transplant if that is necessary. There's only 1 patient that experienced a grade 2 transient infusion-related reaction that resolved without any additional clinical sequelae. But overall, this treatment appeared to be extremely safe with a highly favorable safety profile. So in conclusion, from our initial 9 patients that we have over 1 year of follow-up, we're showing comprehensive efficacy in multiple patients, with at least 5 of these patients having sustained vector copy number, increased bone marrow CFC MMC resistance in quite high ranges, and showing a nice stabilization of their blood counts. We are excited to continue to follow these patients and hope that they do not need additional interventions. And then again, we have additional patients with shorter term follow-up that we're excited about seeing how they do as well. And we'll be reporting on this in the coming conferences. And then we're continuing to treat additional patients with this therapy as well to provide more benefit to more patients, and ultimately hoping to seek approval for this therapy so that we can then enable its use globally without needing patients to be on clinical trials or to travel for those trials. Again, the safety profile is looking quite favorable without the need of conditioning, and we're hoping to continue to provide this to more therapies, to more patients as a therapy that's an alternative to allogeneic transplant. In our gene therapy, we've been fortunate to be able to get away with treating patients without conditioning, given the uniqueness of the gene therapy treatment in Fanconi anemia. But conditioning is an important piece of allogeneic transplants or gene therapy treatment for other conditions. And so our group has simultaneously been focused on trying to improve conditioning. This is some early data that we published on now many years ago when we started to interrogate the competition between hematopoietic cells to try to understand why we do conditioning in the first place. Through these elegant studies that we did when I was a graduate student in Professor Irv Weissman's lab, we showed that hematopoietic stem cells actually compete with other hematopoietic stem cells. And so based upon this, we've been developing novel conditioning approaches that are not broadly ablative to the entire marrow, but instead are targeted towards hematopoietic stem cells specifically. And by doing this, we believe we can create safer methods that reduce collateral damage to other tissues and are also non-myeloablative. And this is an area that my group at our center here at Stanford has been working on for over a decade. Some exciting data to share in this regard. Most of our efforts have targeted this receptor on hematopoietic stem cells known as CD117 or c-KIT. This is a cell surface protein known to be expressed primarily on hematopoietic stem cells and progenitors, and it's the receptor for the ligand stem cell factor, which is known to be critically important for hematopoietic stem cells. Because there are multiple ways of using antibodies to target cells, we have used them in many ways to target hematopoietic stem cells and to eliminate them via various mechanisms. And we and colleagues have shown that antibodies can be used to eliminate HSCs by blocking critical signaling pathways, directing the immune system to kill hematopoietic stem cells, or poisoning HSCs directly using antibody-drug conjugates. And each of these approaches has shown to be an effective way of depleting hematopoietic stem cells. This is some of the data that I was alluding to, showing that as a proof of concept in animal models, we've been able to show that multiple types of these antibodies can be used to beautifully deplete hematopoietic stem cells. On the top row is use of the original antagonistic antibody that we championed to show that this type of concept was possible, showing that a one-time treatment into immunodeficient animals with this antagonistic anti-mouse CD117 antibody could beautifully deplete HSCs with 98% depletion and enable enhanced engraftment without the use of any other genotoxic agents. And subsequently, when I was a postdoc out in Boston working with Professor Derek Rossi and Professor David Scadden, we showed that an antibody-drug conjugate could be used in a similar capacity using far less drug with an even more robust 99.9% depletion of HSCs, and now even higher engraftment post-transplantation with the use of even fewer cells. So multiple ways of doing this now, showing as a proof of concept that depleting HSCs enables robust engraftment without the use of genotoxic agents. And then excitingly, we've really shown that these agents are quite safe and well tolerated. With the first generation antagonistic antibody at high doses, we do see transient graying of skin due to melanocytes. That's an issue that we're seeing. And then with the drug conjugate, we've seen mild transient elevation of the liver transaminases, but none of the other issues that we see with classic conditioning. And just on this photo, you can see that the mouse treated with the antibody drug conjugate looks much healthier and happier and totally normal compared to a mouse that's received a half dose of lethal irradiation. And all of the other tissues that we've assessed have appeared to be quite untouched. Again, others have now continued to build upon our work, adding additional agents to our original antibody, as well as looking at other receptors and targets. And there are three main efforts that are now translating these approaches to patients that have reported on data in non-human primates and now taking this to patients directly. The first generation approach is an antagonistic antibody derived from our mouse efforts that now targets human CD117. This is an antibody that was initially known as AMG 191 from Amgen and has been renamed JSP 191 and is now being developed by Jasper Therapeutics. The second generation approach combined the anti-KIT antibody with a CD47 blocking antibody, again activating the immune system to kill HSCs, and then the antibody drug conjugate approach is being championed and developed by Magenta Therapeutics. The first antibody is now in patients for a number of different indications in severe combined immune deficiencies as a standalone agent, in AML and MDS in combination with total body radiation and fludarabine, in sickle cell disease in combination with total body radiation, Camppath and sirolimus, and then I will talk a little bit about our work in Fanconi anemia here today. And the antibody drug conjugate approach thus far is being used as a treatment only in a dose escalation study in AML and MDS, but we're very excited about all of these approaches and the impact they may have on transplantation for the field broadly, but especially for Fanconi anemia due to the DNA repair deficiencies in these patients that make them particularly sensitive to the current genotoxic agents that we use today. As I mentioned, we've focused a lot of our work on Fanconi anemia, given the high unmet needs in this area and the need for improved treatments. And we've launched into a clinical trial using that first generation antagonistic anti-CD117 monoclonal antibody in this trial that is being led at our center, sponsored by my colleague Matt Porteous and led by my colleague Rajni Agarwal in collaboration with my colleague Alicia Pertina. We opened this trial at the end of last year and have treated 2 patients to date and have reported on excellent outcomes in these patients that will be further shared at additional conferences in the future. This trial combines complementary advances in the field that have enabled us to optimize the treatment of all patients, but especially Fanconi anemia patients, by combining them all into one trial. One thing that we're doing in this trial is optimizing the hematopoietic grafts that we use. So rather than using whole bone marrow or mobilized peripheral blood that is either unmanipulated or CD34 enriched, instead we're using TCR alpha beta CD19 depletion of these hematopoietic grafts to decrease the GVHD triggering T cells and to enrich for stem cells and other innate immune cells that can help protect against infections. And then we've been also combining this with the anti-human CD117 JSP191 antibody to eliminate the use of irradiation and busulfan as part of the conditioning regimen, and combining these two complementary advances with close drug monitoring to enable us to optimize the use of the various drugs in the conditioning regimen, including the JSP191 antibody and the immune suppressants that we use to prevent immunologic rejection. And so this is just a schematic of the trial showing the use of this anti-CD117 antibody in combination with non-irradiation-based immunosuppression, including ATG, fludarabine, cyclophosphamide, and rituximab, taking the backbone of what is used in Fanconi anemia patients for immune suppression today, and again, eliminating the use of the most genotoxic of the agents, including the TBI or busulfan used in many protocols, and replacing that with the antibody that we've developed here, and again, combining that then with close monitoring of all of these drugs and these optimized TCR alpha beta T cell CD19 B cell depleted hematopoietic grafts to improve immune reconstitution and decrease graft versus host disease. And so that is just a bit of information about the novel trials that we're running here at Stanford Children's. We think that Fanconi anemia is a grievous disease that needs improved treatments, and we have been fortunate to work with novel therapies that we think can make a difference for these patients and subsequently others, especially due to the fact that FA is a systemic cancer predisposition syndrome. These patients really need new therapies that do not include genotoxic conditioning treatments. And we've shown that we can do that now with autologous gene-modified cells, as well as allogeneic transplants with antibody-based conditioning. Our group is also continuing to study bone marrow failure in these patients and others to try to understand hematopoiesis better, to further be able to develop improved therapies that might be beneficial to the hematopoietic components of this disease or others, and would love to collaborate with others in the field that have an interest in this area and have access to patients and to samples. And we're very excited about pushing our innovative treatments into other regions of the world so that more patients can benefit from them as well. I just want to quickly acknowledge the many different people that have been involved in these efforts, from my mentors throughout the world, as well as our research group, and the many different individuals that support our efforts, as well as our collaborators and funding agencies. And this is just a list of some of the key people that were involved in these studies.